backbone of NCC, NCC, NCC. And you should also know protein structure, with the confirmation here, primary structure, secondary structure, tertiary structure, and quaternary structure. You should be able to understand all these details that I went over, okay? And I just got to start on lipids, right? Lipids, and also for each of these structures, it's real important is also to understand what molecular forces hold and stabilize it, okay? Obviously, for primary structure, that's covalent bond, that's peptide bond between amino acids. But for alpha helices, beta cleavage sheets, that's hydrogen bonding between the backbone components, okay? The polypeptide, tertiary structure and quaternary structure between R groups, okay? Various types of forces here, hydrophobic interactions, hydrogen bonding, disulfide bridges, and ionic bonds, okay? These are all part of that. All right, so denaturation, okay? Lipids. Well, where did I stop anyway? Did I actually get to lipids?
Yeah.
Yeah, I did.
Back to this one.
All right. Okay. Okay, lipids. Okay, 3 different groups of lipids I'm going to be talking about. One of these, first of all, just wanted to know building blocks, glycerol and fatty acids. Okay, so glycerol is a 3-carbon alcohol, and so what is important is those hydroxyl groups, because whenever we find any sort of linkages between basic building blocks, it always involving these functional groups. Okay, it's not from a simple carbon covered with hydrogen. Okay, so this is a long chain, and I gave that formula that all these fatty acids can be a little bit different in their length and also in their saturation. Okay, so what do I mean? I think this is where I'm starting. Okay, what is a saturated fatty acid versus unsaturated? Okay, a saturated fatty acid has no carbon-carbon double bond. In other words, the fatty acid chain, which is just hydrocarbon except for the carboxyl group at one end, it is just a long chain of carbon atoms, and it is carrying the maximum possible amount of hydrogen. Okay, the maximum amount of hydrogen. There are no carbon-carbon double bonds, and this is said to be saturated. Okay, if it's unsaturated, then we have carbon-carbon bonds. All right. For example, if this is our fatty acid, okay, and we take the 2 hydrogens I have there in yellow, remove them, carbon has to have 4 bonds. Okay, and so to be able to have a stable molecule here, we form a carbon-carbon double bond. Okay, now this is not carrying the full amount of carbon, all right, or the full amount of hydrogen. It's missing some hydrogen, and so this is mono unsaturated, but we can also have things that are poly unsaturated. Okay, where there's more than one carbon-carbon double bond. All right, polyunsaturated, and what is significant biologically about these carbon-carbon double bonds is that this produces a kink in the chain. Okay, now the carbon bonds, since it's tetrahedron, I've been saying that the bond angles are about 104 degrees. Actually, it's 109.5 degrees. That's something I'll never ask you, but nonetheless, that carbon chain is sort of up and down, up and down, up and down, but its general tendency is still a straight chain. Okay, it's relatively straight. But by putting in this carbon-carbon bond, for one thing, it creates the possibility of geometric isomers. Okay, we can have geometric isomers as a possibility, and also it puts a kink in the chain as long as it's in the cis form, not the trans. So it's a cis fatty acid, which in nature, most fatty acids are cis. And I'll explain cis and trans here in a moment. Trans fatty acids are mostly have been made by the food industry. Okay, now we don't try to keep those out of the way, but there's not too many of them that happen to be there. Now I mentioned stearic acid, oleic acid. Both of these have 18 carbons, but what makes them distinct is that stearic acid is saturated. Okay, this is an animal fat. okay, comes, find pretty abundantly in, or fatty acid. It's pretty abundant in animals, whereas oleic acid is unsaturated. And even though they have the same number of carbons, just that little difference means we have to name them differently because their properties are not the same. Okay, so this is a space-filled model where the red is oxygen, white is hydrogen, and all the black are carbon. So this is zigzag, 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 but it's still relatively straight. Okay, so that is a saturated fatty acid. Here we have a cis unsaturated fatty acid. You have carbon-carbon double bond right here. And now this is not straight. Puts that kink in there, okay? And and that has some biological significance. This is a cis because our carbon-carbon double bond is right there. And when I went over geometric isomers, I mentioned that this is the reference point, okay? That axis through the carbon-carbon double bond. And since these two items, the acyl chains, are on the same side of this carbon-carbon double bond, that's cis. Cis means same side, okay? Same side. Whereas these hydrogens, that's not our reference. We're using the carbon chain as the major reference here, okay? So this is cis. If it were trans, this would still be relatively straight, okay? So these are geometric isomers, okay? This is made in the food industry or in the lab. It really doesn't exist in nature. Oleic acid does, but this is the cis form because there's our carbon-carbon double bond and our two acyl chains. Okay, that's abbreviation for fatty acyl. Okay, this is just abbreviation for fatty acid. So two fatty acyl chains, both of them on the same sides, that's cis. This is on opposite sides of the carbon-carbon double bond, therefore that's trans. Okay, so this is no better, or it might even be worse, than saturated fatty acids when it comes to being a problem with cardiovascular diseases. One of the problems with fats, that we'll get to fats in a moment, is the fatty acid. Fatty acids that are saturated or trans tend to clog up arteries, okay, and lead to plugging them up, leading to cardiovascular diseases. Okay, now, there's a lot of variation here in the fatty acids, and let's change this instead of formula for designating fatty acids. I want it to be, let's say saturation formula, because we have molecular formulas, all right, and then we have structural formulas. This here, which shows all of the positions of all the carbons, the hydrogens, and oxygens, and so forth. That is a structural formula, whereas the molecular formula just tells you how many atoms of each element happens to be in the molecule, as I mentioned before. But this here have a saturation formula. So there's a standard for talking about these. For saturated fatty acids, usually we would use two numbers that are separated by a two dots, okay? Now, what the first number stands for is how many carbons are in the structure. Okay, the second number, past the colon, it stands for how many carbon-carbon double bonds there are. Okay, so this here, 16 carbons is palmitic acid. You don't have to know the name's palmitic acid. I'm putting it up here just for reference, okay? This is the biochemical name for it. This is a trivial name, and a lot of times in your textbooks, when you go into biochemistry later on, they will be using a lot of trivial names. But in hexadecanoic acid, that is the biochemical name for it. And I will never ask you about that, okay? Likewise, seric acid, already mentioned that. It's 18 carbons, no carbon-carbon double bond. And it's octadecanoic acid is its scientific name. For unsaturated fatty acids, we still use the same standard. Two numbers separated by a colon. First number is how many carbons there are. Second number is how many carbon-carbon bonds. However, it's also important to know where those carbon-carbon double bonds happen to be. because if we move it, for example, here, if I had, instead of at carbon 9, I put the carbon-carbon double bond at carbon 10 or somewhere else, then that would be a different molecule. Okay, so this is specifically oleic acid. The del in parentheses, so this says that there's one carbon-carbon double bond. And the delta 9 says that that is at carbon number 9, counting from the carboxyl carbon. Counting from the carboxyl carbon. So here's the carboxyl carbon. There's 1, 2, 3, 4, 5, 6, 7, 8, 9. There's where our carbon-carbon double bond is, between 9 and 10. Okay? Get a little bit more complicated. Let's look at linoleic acid, which is called int 912 octet. Forget about it. Okay? Stick with linoleic acid. I can pronounce it, but I have to sit down and think about it. Okay. So here it has two carbon-carbon double bonds. That's what the 2 stands for, at carbon 9 and 12. Okay, so right here, there's our carboxyl carbon. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And you notice that at each one of these carbon-carbon double bonds, the chain bends. Okay, and we can get even more complicated. 22, 6. This is docosahexaenoic acid. It's also abbreviated as DHA. I can say that, okay? All right. And this is a very important fatty acid for neurons, okay? Plasma membranes and neurons. And it has a lot of kinks in it, okay? Notice here, it is 22 carbons long. It has 6 carbon-carbon bonds, and this is where they are located, counting from the carboxyl end. And so it's really kinky, right? Now, in health-wise, you hear about omega-6, omega-3. This is what dietitians use. And what does that mean? This is counting from the methyl end. Okay, biochemists usually would count everything from the carboxyl end. Okay, so when it comes to talking about omega-3s, in fact, omega is the last letter of the Greek alphabet. So we're going to the other end, the last carbon in the carbon-carbon chain, and counting back from the methyl end to where the first carbon-carbon bond has to be. If it is an omega-3, then this would be 1, 2, 3. Okay? The first carbon-carbon bond from the methyl end is on the third carbon, counting back from the methyl. That's what makes it a omega-3. Omega-12s, once again, from here, 1, 2, 3, 4, 5, 6. This here is an omega-6. Okay? All right, so I put that down, omega-9, omega-6, omega-3. All right? Because I still want you to be able to locate the carbons from both ends. Okay, now when it comes to the omega number, the dietitians are not worried about anything beyond that first carbon-carbon bond. Okay, and like biochemists, we want to know them all. Okay, we want to know where all the carbon-carbon bonds are. Because if you move them, you change them. Okay, now why is this so important for dietitians knowing these two? Is that animals usually cannot make carbon-carbon double bonds at the number 3 and number 6 position. So we do have some fatty acids, such as linoleic acid, that we have to get from ready-made in our diet because we can't make it. And so a lot of our omega-6s and omega-7s, we have to get them in our diet. And after that, other types of fatty acids that are unsaturated can be those precursors. Okay, we have some limitations. All right. Okay, so this is the building block of at least two of the macromolecules. So talk about the macromolecules, fats. All right. Now, very often people, dietitians and other people will use fat and fatty acid interchangeably. They are not actually interchangeable. All right? Because the fat is a macromolecule. The fatty acid is a building block of that macromolecule. And so these go by other names. So when it's a fat, It is a molecule that has glycerol and 3 fatty acid chains, okay? These fatty acid chains are covalently bonded to the hydroxyl group. That's where we have the still dehydration reaction, but it happens at the hydroxyl groups and the carboxyl group. Carboxyl group of the fatty acid, hydroxyl group of the glycerol. This is where the dehydration reaction can happen. So maximally, you can have 3 fatty acids attached to a glycerol because it has 3 hydroxyl groups, right? Okay, and this is through dehydration reaction. Now the type of linkage that's produced there is not the glycosidic linkage, it is an ester linkage. And I'll show you the ester linkage momentarily. All right, so this is a maximal amount, and so the good scientific words to use instead of fat is triglyceride. Another word, these are 2 synonyms, triacylglycerols, that's also true too. Okay, so triglycerides, if you look on your food packages, you'll sometimes find that word, triglycerides. Okay, so this is, but we can have, so if we have 3 fatty acids attached to the glycerol, it's a triglyceride, but we can also have diglycerides, okay, where only 2 fatty acid chains are attached, or even a monoglyceride, where there's only 1 fatty acid attached to the glycerol. Okay, they also exist. All right. Now these carbon chains, they can be fatty acid chains. They even on the same triglyceride, the 3 fatty acid chains do not have to be the same length. They can be different. They can also be of different saturations. Okay, some of them might be unsaturated, some of them saturated. Therefore, it can make for a lot of variety for triglycerides. Okay, so this is a figure from your book showing what we're doing here. There's our glycerol. Here is our carboxyl group. Or whenever you see that, it means the rest of the molecule. Okay, it's the rest of the molecule, and who knows, it could be quite long. And so when we have this reaction here, it's taking dehydration reaction, the hydroxyl group from the carboxyl group on the fatty acid, that's where the OH is stripped off, and the hydrogen from the glycerol, and we form that linkage, which is this down to here, that whole box. Okay, in fact, showing writing, well, no, it's not really showing it there, but this oxygen, carbon, double oxygen, that is the ester linkage. Okay, ester linkage. And this is the space-filled model. Now in this case, this fatty acid is completely saturated. In fact, here this figure is showing 3 saturated fatty acids. Okay. Okay, now, triglycerides from animals tend to be solids at room temperature, whereas triglycerides from plants tend to be oils. So peanut oil, corn oil, sesame oil, that's a triglyceride, okay? It has the same types of structures here. When you cook beef or hamburger, when you take out the meat, there's this liquid there that if you let it go to room temperature, it's usually a white, soft solid, okay? These are still triglycerides, right? But why the difference? Why the difference, okay? Because of saturation levels of fatty acids on those triglycerides. In animals, they tend to be saturated, whereas on plants, oils, they have unsaturated fatty acids attached. Now, why does that make a difference? Why does it make a difference? Okay, this is called lard, whereas these are called oils, but still, these are triglycerides, all right? These just happen to carry a lot of saturated fatty acids. These have unsaturated fatty acids. Okay, so why is that? Well, there has to be some sort of molecular force that's holding these together, and that's mostly hydrophobic interaction. Okay, fatty acids are hydrophobic, right? Okay, there's no oxygen except for up there near the glycerol, but the fatty acid chain is just carbon and hydrogen. And for molecular hydrophobic interactions to hold things together, they have to be close packed, okay? So if I have here a fatty acid chain and it's straight, fatty acid chain here it's straight, and they are close together, we have a long piece of surface area that we can have hydrophobic interactions, therefore they stick together, okay? And the strength of the hydrophobic interaction is proportional to the surface area in contact, okay? It's proportional to the surface area in contact. If there's only a small bit of surface area in contact, then that's going to be very weak. It's going to be somewhat fluid. Whereas if we have a long stretch of hydrophobic interaction, it's going to be much more sticky. Okay. I need 2 volunteers to come down here and be fatty acids. Anyone want? Someone come on down. Come on down. Yeah, go. Okay, so I want you both to come here, stand right in front, right next to each other, shoulder to shoulder, okay? These are fatty acids, okay? These are triglycerides. Up here is the glycerol, okay? Here is one fatty acid chain, fatty acid chain, fatty acid chain, 3 of them. And they are saturated. So we have all this area, okay, get real close, right? Yeah. All this area right here where we have hydrophobic interaction, they stick together. But now I'm going to hit it with this, okay? I will, okay. You're going to, we have kink, saturation, all right? Or unsaturated, all right? So that pushes the fatty acids away. Now there is hydrophobic interaction right here, but it's not very strong, all right? So this is too far away, and that's too far away. Molecular forces or molecular bonds are not like love. You can't have it from way across the room. They have to be real close together before there's any sort of action whatsoever. And so this is too far apart. Okay, they're fluid. They can move. Okay, dance, move. Okay. All right, you can go back. Give them a hand. All right, so it's that hydrophobic interaction that makes all the difference. And so because of the levels of unsaturation in oils, they tend to be not stuck together very well. Therefore, they're liquids compared to, they don't close pack, all right? All right, that's the difference there. Okay, so what are some of the functions of triglycerides? Okay, some of them, long-term energy storage, okay? Long-term energy storage, as I mentioned before, the major fuel for cellular respiration to create ATP in the body is glucose. Okay, that's primary. But when you're you can only store so much of it. Okay, about 24 hours supply if you're not eating. Okay, in your liver and skeletal muscle. That's all. Once that is used up, then you have to switch to fats. Okay, and in fact, your liver's capacity is limited. Okay, so if you ate a lot of stuff, and it doesn't matter whether it's protein, carbohydrate, or fats. If you exceed the capacity of the liver and skeletal muscle to store glycogen, then all that's going to be converted into fatty acids and into fats and stored in various places in the body. Okay, now this is a good storage molecule for long-term energy because 1 gram of fat has about twice as much energy as 1 gram of carbohydrate. And so it's lightweight. Therefore, if you stored all of your carbohydrate or all your fat as carbohydrates, you would weigh quite a bit. Okay, so this is convenient. So long-term storage. Furthermore, it's also insulation. A lot of animals live in cold waters. For example, seals, walruses, and so forth, whales. Right under the skin, they have subcutaneous layer of thick layer of fat because this preserves and prevents the loss of heat from core to body. So it's good insulation, and it's also usually stored in strategic positions so that it's cushioning as well. Okay, that's why your rear end is well padded when you're sitting down. Okay, because that's one place where it's good. All right. Also, in some cases, it's also water repellent because it is hydrophobic. All right, so it can be triglycerides can do that. All right, that's one group. Fats, triglycerides. second macromolecule is called phospholipids. Okay, so if we took a triglyceride and removed one of the fatty acid chains and attached to it a phosphate group, okay, phosphate group, and to that phosphate group we attach another polar molecule of some sort, then we have a phospholipid. Okay, so here we have our glycerol and our two fatty acid chains. One end here we have phosphate, and then some other polar group here. And in fact, this is not only polar, it's charged. Okay, and of course phosphate is charged as well. All right, so this creates something that sort of has a dual personality. One end is hydrophilic. It likes to be around water because we have all that polar material. And the two fatty acid chains, which we call fatty acid tails, they don't like to be around water. So this is emphatic, which means that one end will dissolve in water, the other end, the fatty acid tails, are repelled by water. Okay? So we refer to this as a hydrophobic head, and this is the way in which we frequently will use them in a cartoon. There's our hydrophobic head, and we need to understand that that's glycerol, phosphate, and the polar group. And these are the fatty acid tails. All right? So hydrophobic head and long hydrophobic tail. In fact, there's two hydrophobic chains there. All right? So when this is mixed with water in an aqueous solution, those hydrophilic heads want to be around water. They're attracted to water. They're soluble in water. Whereas the tails, hydrophobic, they want to be away from water. And so spontaneously, if you put a bunch of phospholipids in water, they will take on one of two different configurations. Okay? One is that they can become what is called a micelle, sort of a little sphere where all the hydrophilic heads, which want to be close to water, are on the outside. And all the hydrophobic tails are on the inside, where they are sequestered away from water. Okay? So there are structures that are micellar, almost, they're related to micelles, but they're a little bit more complicated than that, for carrying cholesterol and fatty acids through the blood. Okay, because cholesterol and fatty acids will not dissolve in water, and therefore they're hooked to proteins to carry them, but usually they take on sort of a form like a micelle. Okay, it's a little bit more complicated than that. Or else these will become liposomes, in which there are, if we have enough fatty acids, and it also determined by the length of the tails, they will form what is called a liposome, which is still spherical, but it's two layers of fatty acids, back to back. In the middle, we have water. On the outside, we have water. And so we have here sort of a thin skin where there are two layers of phospholipids that are opposite each other. So that in the middle, okay, we have the one inner bilayer where all the hydrophilic heads are in contact with water, and the tails are in the middle, okay, of that bilayer. And on the outside, we have water out here, it's aqueous, and those hydrophilic heads are on the outside. So everybody's happy. The hydrophilic heads are in contact with water, and all of the hydrophobic tails are sequestered away. Okay, they're in the middle. Okay, this is called a liposome, and if we take this and make it real big, then we will have a structure that is a phospholipid bilayer. Okay, but these ends are not free. Okay, so if we take this liposome, which is small, and make it real big, okay, a real big structure, still have water on the inside, water on the outside, we have no free fatty acid tails touching water, okay? And this is the structure, primary structure of biological membranes. So biological membranes are phospholipid bilayers, okay? So what the cell membrane, also called a plasma membrane, is a thin layer of phospholipids around the outside, but it's more complicated than that, as we'll get to a little bit later when we get to cells. It's not just phospholipids, there's also a lot of proteins associated with it. But if we have no proteins, all we have is phospholipids, put this in water, we would form these structures. Why? I mean, without any input of energy, okay? It forms spontaneously, and it's driven by water. Water molecules are very strongly attracted to each other, and they exclude the fatty acid tails. And so this is the most stable configuration that phospholipids contain. Okay, so major importance of phospholipids is that they are major constituents of biological membranes. And we'll talk about biological membranes extensively in the next lecture. Next two lectures, actually. Okay, third group is sterols. Now, the basic building blocks for sterols I'm not going to go into. Yes?
Is there water inside the center of the lipid node?
Yeah. So there's water inside and water outside, because we have to have something on the inside. Okay, so yes.
Did you say all three of them are these three structures?
What's that?
Did you say all three, the particular hydrogen and my cells are the
Yeah.
so
Well, phospholipid bilayers are. This does have a phospholipid bilayer. Okay, I want to point that out. But why do we make a special mention of liposomes? Is because they're relatively small, whereas the cell membrane is, in comparison to that, relatively big.
and all of them are stable.
Yeah. Yeah. Okay. Okay. Now, sterols. I'm not going to give you the basic building block of that. It's not glycerol. It is not fatty acids. It's something else. And there are other types of lipid molecules that likewise have different types of building blocks. Waxes happen to belong to this group of lipids, but I'm just going to talk about sterols. Okay, sterols is a hydrocarbon ring. Okay, four hydrocarbon rings closed on itself like this. And although this figure has numbers up here, I'm not going to impose upon you numbering any of the carbons within the structure. Okay, so it is mostly hydrocarbon. Okay, so those rings have carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. It's hydrophobic. Okay, doesn't dissolve in water. Okay, now from these rings, one of the most important ones here is cholesterol. Okay, so this is the typical ring structure for rings, but cholesterol has this tail right here. Notice it's still hydrocarbon, and it has one hydroxyl group over here. Okay, so this is the only thing that's attracted to water. All the rest of it is not. And so the vast majority of it does not like to be in water. It's hydrophobic. Now, why is cholesterol? Cholesterol is real important here because all other sterols are constructed from cholesterol, right? So cholesterol is also, or sterols, not just cholesterol, but sterols are also part of membrane structure. Okay, they're found. Animal cell membranes have a lot of cholesterol. Plants, they have a little bit, but it's not very measurable. They have a different type of sterol called cystosterol. You don't have to know cystosterol now, but you will in the future. Okay, I'll get there. In fungi, they have a different type of sterol in their plasma membranes as well. But cholesterol is pretty abundant in animals. Furthermore, certain steroid hormones, such as estrogen and testosterone, and also a mineral corticoid called aldosterone, which affects your kidneys and the water and salt balance in your body. These all happen to be derived from cholesterol. Okay, so what I need to point out here, homeostasis. You've probably heard a lot about cholesterol. Cholesterol, bad cholesterol. Okay, actually, what you have is homeostasis. Too much is bad, too little is bad. Okay, you have to have cholesterol. Your liver makes it. Okay, your liver makes it. You need it because to make hormones, right? But in our diet, we get, with the types of diets that we get way too much of it. Okay, so homeostasis. Too much is bad, too little is bad. Okay. All right, so that's lipids. So with the nucleotides, I'll be talking more about their structure when I get to heredity, but right now just introduce what they are. So this is the fourth group of biological molecules. These are nucleotides, nucleic acids and their derivatives, and their major function, or at least what most people would think of, first of all, is that they are hereditary material, and in the form of deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. They have other functions too, mostly they're derivatives, and I'll come to that. So let's talk about DNA first. Okay, DNA, it stores information, okay? And it also has certain switches for controlling the expression of that information. And so the functional unit of DNA for the information is called a gene, okay? And so that's a segment of the DNA molecule, okay? It has a switch at the very beginning of it, a termination signal at the end of it, so that gene is sequestered from other genes in the DNA. But the types of information for protein encoding genes is that they determine the primary structure of a polypeptide. So they determine what is the amino acid sequence of a polypeptide. The amino acid sequence determines how that polypeptide folds. It determines, so the amino acid sequence determines secondary structure. It determines tertiary structure and even quaternary structure, okay? So this is very important. And it is the interaction of all these proteins that determine the metabolic process and the structure that happen to be inside the cell, all right? So this is a very important molecule. Now, and it is a polymer, and its basic building block, the monomer, is something called a nucleotide. Nucleotide has three components to it, okay? It has a aldopentose sugar that's in a ring form, an aldopentose sugar that's in a ring form, and it has a phosphate, okay? And it has a ring structure that is called a nitrogenous base, okay? So I'll get to these nitrogenous bases in a moment. The nitrogenous base is attached to carbon number 1, okay? Just practice. There's our ring closing oxygen, right? Okay, and so now this makes it a little bit harder to find the anomeric carbon, because right here there was once upon a time an OH group, okay? That's what the nitrogenous base is there. This is carbon number 1, 2, 3, 4, 5. So the nitrogenous base is attached to carbon number 1, and the phosphate is attached to carbon number 5. Okay, so this is an aldopentose. And so this whole thing together is the basic building block of nucleic acids, a nucleotide. All right, so let's see here. Okay, now there's only two types of sugars that are involved in DNA and RNA. Okay, this is deoxyribose. It's a shame you can't see the lines. It's yellow there, but nonetheless, that's a ring structure. It's carbon 1, 2, 3, 4, 5. There's our ring closing oxygen. There was our OH group. This is another derived monosaccharide. This is a normal simple monosaccharide, ribose. This is ribose, okay? Carbon 1, 2, 3, 4, 5. I've mentioned for derived monosaccharides, acid monosaccharides, amine monosaccharides, and now we have a third type of derived monosaccharide called a deoxy monosaccharide. Okay, why is it called deoxy? Except for the carbon, anomeric or carbonyl carbon, every carbon should have a hydroxyl group in the linear form. But look at carbon number 1 here. There's our hydroxyl group. But carbon number 2. Carbon number 2 on the deoxyribose, it has no hydroxyl group. In other words, it's deoxy. D meaning no, taken away, it's not there. Oxy is referring to oxygen. So the number 2 carbon doesn't have any oxygen there, all right? And so this makes it different. These two have different shapes, and therefore enzymes and other structures in the cell that can recognize DNA will not necessarily recognize RNA. Therefore, they play completely different roles. This is the sugar that's in the nucleotides that compose DNA. Ribose is in the nucleotides that compose the macromolecules for RNA. RNA is ribonucleic acid, DNA is deoxyribonucleic acid, okay? All right, so that nitrogen base, it is an organic ring that's in its backbone. It has nitrogen and carbon, okay? Now, we've come across amino acids, they have nitrogen, all right? But you'll be able to recognize them, though, by the NCC backbone. But with these, we have a lot more carbon, or a lot more nitrogen. So some of these form just only a single ring, and they're referred to as pyrimidines. And there are many different types of nitrogen bases in the body, but as far as being formed in the nucleotides that form RNA and DNA, there's only 5 different bases, nitrogen bases. Okay, there's uracil, thymine, and cytosine. These are pyrimidines. They consist of just one single ring. Okay, and notice in the backbone there is nitrogen, carbon, 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 nitrogen, and so forth. If they have double rings, they're called purines. And the only ones that we find in the hereditary material is adenine and guanine. Okay, have double rings. These are nitrogen bases. Okay, all right. So what the difference between DNA and RNA is both in the sugar that compose the nucleotides. DNA has deoxyribose, and it has no uracil. It has thymine. Okay. In fact, if you look at thymine and uracil, I would never have you draw these, but if you see things like this on the quiz, you should recognize them for what they are, as pyrimidines and purines, nitrogen bases. Okay. But if you look at thymine, the only difference is between thymine and uracil is right there. This has a methyl group, thymine acid, and therefore in RNA, you would have uracil instead of thymine. In DNA, it has thymine instead of uracil. Otherwise, they have all the other bases. Okay? So when we show the sequence in DNA, we usually are showing just only symbols for the bases. Okay? Because in DNA, the sugar is always deoxyribose, always deoxyribose. In RNA, it's always ribose. The only difference between nucleotides is what nitrogen base they have. And therefore, A stands for adenine, and C for cytosine, G for guanine, and so forth. And those are the symbols we use. Okay, so putting these nucleotides together, we form polynucleotide. Okay, it's a chain. Okay, so it's polymerization, and once again, we form a special type of linkage. Okay, this is called phosphodiester linkage, and it's formed by dehydration reactions. Always, the hydration reaction is between the phosphate on carbon number 5 of one nucleotide and the hydroxyl group on carbon number 3 of the next nucleotide. Always. And that forms carbon, phosphate, carbon. That is the phosphodiester linkage. Phosphodiester linkage. Okay? And so these molecules have polynucleotides. They have directionality. They're polar. They're polar. If we'll start up here at this phosphate, and we're walking down the backbone through the sugar, we would first of all go into 5'. Okay, why prime? Because the carbons on the bases are also numbered. And this, by saying prime, we know that we're talking about the numbering of the sugar carbons. Okay, and therefore we would go from phosphate to 5, 4, 3 into a phosphate, 5, 4, 3, 5, 4, 3, 5, 4, 3, and therefore this direction going from top to bottom is referred to as, we need to know the direction. It's important, as we'll see a little bit later. 5', 3'. Okay? And there's a reason why I'm saying 5' to 3', emphasizing that instead of 3' to 5'. Okay? Because I'll give you a hint, well, I won't give you a hint, I'll just simply tell you, is that the processing and way that the chains are put together is always in the 5' and 3' direction. Okay? All right? Okay. Furthermore, the DNA molecule is not just one polynucleotide. It's two polynucleotides that are intertwined with each other. Okay? And so this is what makes it a double helix. Okay? The double is referring to one strand. We call these chains, it's a polynucleotide, but we use also the word strand. Strand. Okay? So this is one polynucleotide, and there's another polynucleotide. Okay? They are right next to each other, and there's hydrogen bonding between the two strands through the bases. The bases are hydrogen bonded. And when this is in water, because of the bond angles, they all form a spiral. So this is what is a helix. So don't confuse this with an alpha helix. Okay, that's a structure that's in proteins. But this here, double helix, DNA, double, and there's two polynucleotide strands, two polynucleotide molecules, and held together by hydrogen bonding, and they're in a helix. Okay, and so this is the structure of DNA. Now, notice here too that 5' to 3' on the polynucleotide on the left here is going down, whereas 5' to 3' on the other one is going up. These two are going in opposite chemical directions. Opposite chemical directions. And this is referred to as their being antiparallel. Antiparallel. They're going in opposite directions. One's going 5' to 3' down, the other is going 5' to 3' up. Okay? Okay. RNAs are just one polynucleotide. Okay? They consist of one polynucleotide. And there's many different types. We'll come across a lot of different types of RNA. Some of them that we'll come across relatively soon, messenger RNA, ribosomal RNA, but there's more. Now, although these are usually just a single polypeptide, not always, because sometimes there might be more, they can take on far more complicated structures. And in fact, they may have some internal complementarity so that within them they can form strands that are hydrogen bonded to each other. With the hydrogen bonding, it's always cytosine, guanine. They're the only ones that will form a bond together. Adenine, thymine. Okay, these are called Watson-Crick rules, base pairing rules, and so you never get A bound with hydrogen bonded to G. Okay, because they're in different positions, and the only thing that we have a stable bond is between A and T and G and C. Notice that A and T will form two hydrogen bonds. G and C forms three hydrogen bonds. Okay, we'll come back to that later. Okay, so this is another space-filled model. This is giving you a better sense of the fact that it is in a double helix. This is from your book, right? Okay, just to summarize here, the differences between DNA and RNA is that DNA is two polynucleotides. The sugar that composes composes the nucleotides is deoxyribose, and it has four different nitrogen bases. That's all. Building blocks just have four different types. It has thymine but no uracil, whereas in RNA it's usually one polynucleotide. The pentose sugar is ribose, and it has uracil replacing thymine. Okay. So in this case, yes, thymine instead of uracil. Okay, so we'll have more to say about DNA and RNA later, but there's also other molecules that belong to this group of biological molecules that have quite some significance, nucleotide derivatives. I mentioned adenine trinucleotide. Okay, that is the energy carrier in the body, the major energy carrier. And it is a derived nucleotide, derived from adenine. Okay, this here is adenine base, ribose, okay, ribose, and on the number 5 carbon, 1, 2, 3 phosphates. Okay, so those phosphates hold them together because those phosphates have a lot of oxygen and even negative charge, okay, that repel each other. There has to be a fair amount of chemical energy to hold those together. All right, so when you split off, hydrolyze one of these, and the phosphate comes off, you now have adenine diphosphate, okay? And the energy of that bond, which is fairly substantial, is released and can be used to do work in the cell, okay? Whether it's muscle contraction or movement or biosynthesis, and we'll see how that's used a little later on. Okay, and this has to be regenerated. That phosphate has to be put back on to make it ATP, because once you take it off, that energy is gone, you use it, and that is the whole function of cellular respiration, is to regenerate ATP. Okay, all right, so this is adenine. Some of these are coenzymes. Okay, when we get into cellular respiration, we're going to come across a molecule that's called coenzyme A. Okay, it is a nucleotide derivative. It's pretty complicated. Okay, so here, once again, adenine. This is was ribose, but notice carbon 1, 2, 3. Put a phosphate there. Then it has all this structure attached to it. Notice at the very end of that long tail is a sulfhydryl group. Now, I don't expect you to draw this or recognize it or anything, but we'll be talking about coenzyme A later, and that sulfhydryl group here will be coming forth. All right. These are coenzymes, and there are others that are coenzymes that I'll mention a little bit later that are nucleotide derivatives. So what is coenzyme? Enzymes, as I said, they're catalysts, biological catalysts, and most of them are proteins, not all of them. But what a coenzyme is that it is a non-protein addition to a enzyme, proteinaceous enzyme, and it's necessary for its function. And we'll cross that bridge again a little bit later. Signaling molecules that we'll talk about this later. I'm not going to show it now. Cyclic adenosine monophosphate. Okay, and it is a way for cells to communicate with each other and even within the cell communication. So these are some of the roles of nucleotides. So nucleotide, this group of biological molecules, nucleic acids, is not just hereditary material. Okay, they can also be coenzymes. They can also be energy carriers. They can also be signaling molecules. All right, and this is just another figure. All right, any questions? So that's nucleic acids, or the 4 groups of macromolecules, and of course, one of the things about biology is that it builds on itself. We will come back to all these again. Okay, we'll build on. Any questions? Yes?
Could you explain 5 prime and 3 prime again?
Okay. Let me go back and get that. Okay, go back to this figure here. In fact, I'll go ahead and make it a little bigger. Okay. Because of the orientation of that sugar, okay, that sugar is what causes this to be polar, okay? Because it's in the ring form, carbon number 1, carbon number 2, carbon 3, carbon 4, carbon 5. Now, if you're starting at this end, it ends with phosphate. You go down here, it ends, if we didn't go down to the next one, it would end with a 3' hydroxyl group. Okay, if you're walking down the backbone, we're going from phosphate, 5' carbon, 4', 3', phosphate, 5, 4, 3, phosphate, 5, 4, 3, 5, 4, 3, 5, 4, 3. That is 5' and 3'. The other side, there's a 5, 4, 3, 5, 4, 3, 5, 4, 3, 5 prime to 3 prime is that way. Okay? Does that make sense? Got it? Yeah.
Why is it prime?
Because the carbons in the nitrogen base, they're number 2. They're number 1, 2, 3, 4. And to make the distinctions that when we're talking about these numbers, because I'm not gonna have you number the carbons in the bases, okay? That's, when you go on to biochemistry, you know, you get to do that, okay? But we're not gonna mess with that here. Because what's important here is that 5 prime to 3 prime through the sugar. So to make a distinction between what, when you're having a nucleotide, what are you referring to by 1, carbon number 1? If it's carbon number 1, it's in the base. If it's 1 prime, it's in the sugar. Okay, so that's what the prime is for. So when I went down here, it's really 5 prime, 4 prime, 3 prime, 5 prime, 4 prime, 3 prime, and so forth. Okay, anything else? Yes.
Can you go over the micelles form?
How the what?
The micelle?
The micelle? Okay, so when you put fat or phospholipids in an aqueous solution, those fatty acid chains are repelled by water. And so you need a structure where the heads are in contact with water and the tails are sequestered away from water. Okay, so one stable form it can have is just have a bubble, okay, a sphere where all the heads are on the outside and the tails are on the inside. Now, if those tails are too long, then they're going to push this molecule too far apart and it's not going to be very stable. And so if the fatty acid tails are long, then a micelle is not stable. And therefore, the better thing there would be a liposome, where you have 2 phospholipid bilayers, where it's aqueous on the inside, aqueous on the outside. Okay, it's sort of like having a basketball where our phospholipid bilayer or volleyball is around the outside, you have air on the inside, air on the outside. Okay, and that phospholipid bilayer. Okay, yes?
Could you go over the ester linkage one more time?
The what?
Ester linkage?
Ester linkage? Okay. You're talking about in lipids. Yeah, okay. Ester linkage is right here. In forming, actually, let me go to one more. Actually, go back right here. This is good. Okay, so here is our glycerol, and here is the fatty acid chain. That R stands for the rest of it. Okay, but what's important is that carboxyl group. Okay, so it's a dehydration reaction, and therefore we're going to remove hydrogen from that and OH from this, and we form this right here. Okay, now this is showing the glycerol, but the ester linkage is that oxygen double bonded and carbon, oxygen, carbon, and it includes the double bonded oxygen. That's what makes it an ester. Okay, an ester looks like this. It has oxygen and double bonded oxygen. That's an ester. Okay, does that make sense?
Yeah.
Okay. All right. Any other questions? Okay. Okay, for number 3, moving on. Okay, so we're going up the hierarchy now. Okay, so what we've been looking at is at the level of atoms, molecules, macromolecules. Now we're going up into cell structure. Okay, so this is an introduction to cell structure, and we'll be talking about cells quite a bit. So I've given you this particular scenario of building one on top of another, and I mentioned that each of these levels have their own emergent properties, okay? But those emergent properties of life, being able to reproduce, being able to sense changes in the environment and respond to it, having metabolic processes that they use energy and transform it, those first occur at the level of the cell, okay? And so everything from the cell up is alive. see them with your unaided eye. In the 1500s and so on, many people started working with glasses to form telescopes and microscopes that magnified things. And so as microscopes improved, an Englishman named Robert Hooke in 1665, he took a little thin slice of just underneath the outer bark of a tree, put it on a microscope slide, and he saw all these little boxes, and he called them cells. And so this was the first person who published anything about structures that become called cells. And the reason he called them cells is because these look like all the little rooms in a monastery where monks happen to live. Okay, because the monk lived in a little tiny room, and they were just adjacent to each other and so forth. And so since that's what he saw, he called them cells. And over the years, as microscopes improved and more people looked at different types of living organisms through a microscope, they noticed that everything consisted of boxes, rectangles, or some sort of bag. Okay, and so in 1839, two Germans, Schleiden and Schwann, they proposed, they took all these observations that had been published up to that time and generalized, made a generalization that all living things consist of cells. And so this gave the cell theory, and the cell theory that they proposed has 4 different components to it, 4 principles. Some of these principles are relatively modern. They're not original for Schleiden and Schwann's cell theory, but one generalization is that all living things consist of cells. And thus far, that has proven to be indeed so, although we have to sometimes expand a little bit our idea of what some cells might look like, and also all cells come from other cells, okay? In other words, cells reproduce other cells, all right? Something that's more a little bit more modern, because they really didn't understand at that time chemical reactions, but all the chemical reactions that animate life happens in the context of the cell, okay? They happen inside of the cells, and furthermore, this is also absolutely a modern addition to this cell theory, is that all cells have hereditary material that contains the information for making similar cells, okay? So these are the 4 principles of cell theory, right? And now, one of the problems, why are they so small, okay? Cells are relatively small, and so if we took a box cube, where each side of that cube has a dimension x, then the surface area of that total, because it has 6 sides, right? So if this dimension is x, and so this here would be x squared, and we have 6 of those, then the surface area is 6x squared, okay? But the volume is x cubed, all right? Because that's x, and so inside, in other words, we have a problem here with regard to surface area and volume. Now, if we double this, okay, actually make it 2x on the side, then our surface area is going to be 24x squared, but the volume, 2x squared, is going to be 8x squared. And you can notice here, if we take the ratio between surface area and volume, this is 6 to 1, but now it's only 3 to 1. And if we make it even bigger, then it's 2 to 1, and so forth. The whole point being here is that surface area and volume don't increase linearly. Okay, and in fact, the surface area increases as a cube of a linear dimension, whereas the volume is increasing at, by 3, by a cube, by square. Sorry, let me back up there. You should always go by what I mean to say and not what I say. Okay, so the surface area is increasing as a square of linear dimension, whereas the volume is increasing as a cube. In other words, volume is increasing faster when you make these things bigger. If you don't change the shape, if you maintain the same shape. Now, why is that significant? Okay, it's significant for this. Cells have to exchange materials with their outside. Okay, they have to take in nutrients. That is a function of the surface area. That's a function of the surface area. Furthermore, any waste products also have to be transported out of the cell. That's also a function of the surface area. Consumption and the production of waste material is a function of the volume. These two have to stay in balance. They have to stay in balance. If we get so large that there's more waste material being produced or a higher demand for resources than what the surface area can deliver, it's going to be fatal to the cell. And therefore, these two have to stay in balance. Furthermore, some of the movement inside of the cell. I will point out that there's mechanisms in cells for moving things very rapidly. However, a lot of molecules just can be moved around the cell by simple diffusion. And the larger the volume, the longer it will take that material to diffuse. And that becomes a limiting factor as well. And therefore, this is one of the reasons why cells are relatively small, because there is a surface area to volume problem here, okay? And so one way, if you're going to increase the mass of an organism, make it bigger, then it's divided up into smaller cells, so that we have something that's multicellular, and all those cells can communicate with each other, but no one cell in this larger structure has a problem with surface area volume ratios. Okay, so the cells remain relatively small. Now, there are some cells that are pretty big, okay, in some cases, but there's a trick to that. They change shape, okay? So if we have, okay, let's just jump to plants. Not there yet, but we'll talk about plants, but this is a good place to mention it. Some plant cells are pretty big, okay? Still have to see them in a microscope, but they can be pretty big, okay? One of the reasons for that is they have this membrane-bound structure called a vacuole, central vacuole. And that's just filled with water. It's inert, okay? All the living cytoplasm or volume where metabolic activity happens to happen is in a very, very small volume along the edges. And so the cell can be big and yet not have that conflict, okay? And there's other tricks to the game as well, all right? But by and large, this is what happens. Now, the way in which cells are studied, of course, originally with microscopes. Today, we have all sorts of biochemical and types of techniques to investigate what cells are doing. But originally, it was microscopes. And so I want to say something about microscopes, because we will have a microscope lab a little later, is that they do more than just magnify, okay? What magnification is an increase in the size of the image that you're looking at, but that doesn't necessarily increase the clarity, okay? So if you took a picture on your phone and you blow it up, blow it up, blow it up, what does it look like? There's a bunch of little dots, okay, pixels. That doesn't increase the clarity. And so microscopes do two things. One, they do magnify, but also they have what's called resolving power. And so what is the resolving power? It is the maximum resolving power is the distance between 2 points where they still appear as 2 points. So if we have a point here, an object there, and an object there, if these happen to be 1 micrometer from each other, and the resolving power, maximum resolving power of our microscope is 1 micrometer, then we would see 2 of those. But if these were closer together, say that this distance was a 1/10 of a micrometer, and our maximum resolving power of the microscope is 1 micrometer, these things would blend together and look as one object, okay? And so that's the limit. Now, one of the things that limits this resolving power, part of it is structure of the glass made for the lens, but one other parameter here is the wavelength of the medium that we're using to illuminate it. So we usually use visible light, okay? Visible light travels in a wave, and therefore the shorter the wavelength, the higher the resolution you can have. But if we have real short wavelength, ultraviolet, that's going to burn your eyes, okay? So we have a little bit of a limit of how far we can go down. So this is limiting. A light microscope that you'll be using in the lab, at most, the best light microscope can give a resolution of 0.2 some larger structures in the cell, but there's a lot of smaller stuff there that the light microscope would not be able to resolve. Back in the 1950s, what was invented was the electron microscope. So instead of using light as the illuminating medium, it used a beam of electrons. Okay. Now that means that the tube in which those electron beam has to be created has to be in a vacuum. Okay, so these are big devices, but because of the wavelength of electrons here as they move as a beam, this can resolve things down to 0.2 of a nanometer. Okay, that's real small. All right, so you need to get familiar with the metric system. Okay, and so an exercise, lab exercise 1, yeah, we'll talk about the metric system, but you need to be able to use it. So these particular devices, so with that resolution, you can see organelles and even proteins, okay, to a certain extent, all right? Whereas with a light microscope, you would some of the larger organelles, yes, you can see, but a bunch of smaller organelles, proteins by no means will you ever be able to see in a light microscope. So there's two different types, transmission electron microscope and scanning electron microscope. This is called also TEM, okay, transmission electron microscope, and scanning, scanning electron microscope, SEM, sorry about that. And they're a little bit different. Okay, so with the transmission electron microscope, you have to prepare a specimen on a slide, and it has to be very thinly sliced, and there's a bunch of techniques. It has to be dehydrated. First of all, it has to be a fixative put on it, so it cross-links proteins and other structures to keep them together, and then it has to be dehydrated, and then coated with a heavy metal, usually gold, very, very small gold pattern. And then it's mounted on here, and up there you have a tungsten thread wire that will produce the beam of electrons. This has to be a vacuum, and the beam of electrons will pass through the specimen, and there are sensors down here that pick up the patterns of electrons. Where there's thick structures on one side, the electrons are absorbed, and where there's open space, the electrons go through. And so you don't look through any lens here. This is put together, the image on a computer screen, okay, so that you can see the pattern. With a scanning electron microscope, it doesn't have to be thin section. It can be pretty big, in fact, okay? But once again, the specimen has to be fixed with a fixative, dehydrated, and then coated with a heavy metal, usually it's a gold powder. And in this case, the beam of electrons bounces off of it, okay? It doesn't go through. Bounces off, and there's sensors around that will pick up the scatter pattern of the electrons, and once again, an image is created on the computer screen, okay? So this is in general how this has to work. One of the disadvantages of this, even though you can see small structures, you can't look at anything that's alive, okay? Because by the time you fix it with a fixative and dehydrate it, it's dead, okay? So even when you see dead material, you can't see live stuff. Whereas in the light microscope, you're going to get to see some things that are alive and moving, all right? Furthermore, there's always this question of structural artifacts, okay? Because you're putting this through some processes, okay? So is the item that you see, is it real? Was it really there? Or is it a product of the fixation and dehydration and so forth, okay? There's ways to get around this, but nonetheless, this is one of the problems with it, okay? All right, so cells. Although there's many, many, many different types of cells, most cells' life have a lot of things in common, okay? So all cells have an outer covering, a phospholipid bilayer that is the boundary of the cell, called plasma membrane. It's also called a cell membrane, so these are synonyms: plasma membrane, cell membrane, okay? They all contain genetic material. Furthermore, they have space, fluid-filled space on the inside that's called cytoplasm. And inside of that cytoplasm are various types of molecules that are dissolved. And they all have workhorse enzymes and proteins. So these are some of the things that they have in common. And when we look at all the different types of cells, like in humans, we have probably well over 200 different cell types, okay? But they all will have these things. And furthermore, if we take all of the cells in life, period, there's two broad categories of cells. Two broad categories of cells that are called prokaryotic cell and eukaryotic cells. So I'm going to give a list here of items. This is a prokaryotic cell. This here, these are eukaryotic cells. Okay? So the distinction between these is that how they pack the genetic material. Okay? How they pack the genetic material is different. Prokaryotic cells, their DNA is on one molecule, one circular DNA molecule. Okay? It's a double helix, but it contains all of the information. In other words, the whole genome is on one circular DNA molecule. So when we say genome, we're referring to the collection of all the genetic information typical of a particular organism, okay, a particular species, right? Whereas in eukaryotes, the DNA molecules are linear, they're not circular, and there's more than one, okay, more than one. And furthermore, that's not, it's wrapped around proteins. They're here, this molecule in prokaryotes has very little protein in it. There is some, but not much. And some of these proteins are called histones. We'll talk about histones a little bit later. But typical, and they're arranged into what are called chromosomes. About half of the matter in a chromosome, half of the mass is a nucleic acid, half of it is protein, okay? Whereas you have nothing like that in prokaryotes. So they package their DNA differently, okay? When I say that they have more than one DNA molecule, more than one different DNA molecule that carries different information, okay? We may have one chromosome that has the genes for skin structure, muscle structure, etc., and another chromosome that has information for blood type and so on. In other words, you have to have all of these separate DNA molecules for the genome, okay? They also differ in a structure, an organelle called the nucleus. Prokaryotes don't have a nucleus, whereas eukaryotes have a phospholipid bilayer forming a nucleus, an organelle in which most of the DNA is housed, okay? So the DNA in eukaryotes is inside of the nucleus, whereas the DNA inside of the prokaryote is in the cytoplasm, okay? It's out there. All right, now, the Greek word karyon referred to grains. It could be a sand grain, it could be a grain of wheat or so, and we have opted that, we've taken over that word to refer to as a nucleus. So we've seen the word karyon, we're referring to a nucleus. The pro is, in this case, means before. So the before the evolution of a nucleus. And we have good evidence that prokaryotic cells evolved first. Eukaryotic cells came later, okay? EU means true nucleus, okay? So these have a nucleus, those don't. Furthermore, within the cytoplasm, there have prokaryotes, there's very few membrane-bound organelles. What I mean by membrane-bound organelle, it's a structure in which there's a phospholipid bilayer that composes an organelle, okay? They don't have it. They have a few, some, but very, very few. Eukaryotes, they have many. And we're mostly going to be talking about those eukaryotic organelles. Many of them that are surrounded by a membrane, biological membrane, phospholipid bilayer, and each of these organelles have specific functions, okay? And that's what we have. Now these are the characteristic differences between eukaryotes and prokaryotes. This is a physical property that generally holds true, but not 100%, is that prokaryotic cells are usually small. They're on the order of 1 to 10 micrometers. There's exceptions. And eukaryotes are usually larger. Okay? Once again, about 10 times larger. Okay, this is just rough averages, all right? And furthermore, all the life on Earth, there's evidence, we'll get to this evidence a little bit later on. There's 3 major branches to the evolutionary tree, which we call domains. So we have the last universal common ancestor of all life, and even though we have no fossils of it, based on the structures of living cells, we can deduce what that last common ancestor had, okay, minimally. And so from that, there were 3 branches. One, eubacteria. Archaea. And eukarya. These are the 3 fundamental, deep branches of the evolutionary tree. And they're called domains. And so, eubacteria and archaeans, they are prokaryotes. They consist of prokaryotic cells, whereas eukarya, as the name shows, it has eukaryotic cells. And so this includes fungi, algae, protozoans, animals, plants, okay? Typical types of things that we know of, okay? All right, this is time to stop. So this is where I'll stop here and